Welcome back, everyone. We are in the middle of talking about transcription. We just discussed some different types of RNA compared what their functions are, and we discussed those four main steps of RNA transcription. We have the RNA polymerase binding to the DNA. We have initiation of transcription. We have elongation of your DNA, of your RNA strand, and then you have termination of the whole process. So we know those four main steps. Now we're going to get into discovering a little more detail about each of these steps. So first with binding, RNA polymerase is going to bind to the DNA promoter site. And the DNA promoter site is going to be several dozen base pairs that tell you where RNA transcription needs to start. You don't want RNA polymerase just anywhere randomly binding to anything and starting any random piece of RNA molecule. And when we're talking about this, we're going to use the terms upstream and downstream. So if this is our gene, this is the promoter unit, here is the start site. This is the first nucleotide that gets transcribed, and then transcription proceeds in this direction. So this is the direction that's downstream, following along where RNA polymerase is moving. Upstream means moving up before, opposite to where RNA polymerase is going. So that means our promoter then is upstream. If this is moving downstream, the promoter is on this side. It's upstream of the transcribed sequence. Oh, sorry. So in our promoter, our transcription start site is almost always going to be an adenine, although it's almost always a purine, but typically it's adenine. And about 10 base pairs upstream, we have our -10 sequence. Sometimes it's called a Pribnow box, and its sequence is TAT, AAT. Upstream a little bit more, you have the -35 sequence, which is TTG, AGA. And these two sequences are very evolutionarily conserved. That means when you're looking at diverse species that have very different DNA, these sequences are still the same. That tells you that there's not a lot of room for mutations in this sequence, that mutations don't give rise to new organisms, mutations just can't survive. So these sequences are very important to the transcription of genes. A consensus sequence of a promoter or other conserved sequences, these are going to be when you have nucleotides that are most commonly found in a particular position. And then a lot of bacterial promoters also have an upstream elements or up elements that are further upstream from here. Those upstream elements and consensus sequence are going to be associated with very strong promoters, things that need to be very rapidly and efficiently transcribed. For example, our RNA genes, you need to be constantly making lots of ribosomes. In bacteria, there is one type of RNA polymerase. And it is that single kind of RNA polymerase that is going to transcribe all RNA, whether we're talking about messenger RNA, ribosomal RNA, tRNA, that one type of RNA polymerase is going to do it. And this RNA polymerase has two alpha subunits. Here's an alpha and an alpha. You can see it's kind of binding to like this upstream element. There's two beta elements, a beta and a beta prime. You can see it's interacting here with the -10 and -35 sequences. And the sigma subunit that you can see is dissociable, so it can let go or be bound. If you have the core element, this part without the sigma subunit, it can still work to go through and do RNA synthesis. The alpha and beta subunits have all of the necessary components to carry out RNA synthesis. But the holoenzyme, the word holoenzyme means you have the whole thing, the complete subunit, all the subunits making a complete enzyme. You have to have the holoenzyme with that subunit to make sure that you're binding correctly to these upstream elements, to the promoter sequences, so that you start initiation correctly at the correct spot in your DNA molecule. The sigma subunit especially helps promote binding to the -10 and -35 subunits. There are different sigma subunits that can help initiate different gene transcription. And the alpha subunit interacts with the up elements to enhance promoter binding. And there's a discriminator element downstream of the -10 that is going to help stabilize the RNA polymerase to make it really able to stably bind to that promoter sequence. Once we have binding, the next step is initiation. Initiation is going to involve first unwinding your DNA and then actually beginning to add your nucleotides. On your DNA, you have two strands. One is the coding strand and one is a template strand. So the coding strand, although it's very convenient for us as students to take that coding strand and use it to figure out your RNA, in a cell, really the template strand is what's being used. And when it opens up this template strand, it can take the sequence of the DNA nucleotides here and use that to know what incoming NTP or nucleoside triphosphate molecule, what needs to be added here. So when I have a T on my DNA, I know I'm going to add an A on my RNA. When I have a C on my DNA, I know I'm going to add a G to my RNA. I'm just adding whatever nucleotide is complementary, A's and T's and U's, and C's and G's together. The RNA polymerase is going to catalyze that bond or help those two NTPs, those two nucleotides, bond together. And it is going to then, by bonding the different RNA nucleotides, start creating an RNA sequence. It doesn't need any promoter. It doesn't need any primer like we saw in DNA replication. It can just start at the promoter and start binding without needing a primer there. RNA polymerase is going to keep initiating sequence until you get a chain that's about 9 nucleotides long. And then it's not going to move along. It's going to keep repeating, making these small little nucleotides up to 10 nucleotides long. And that's called abortive synthesis. It's going to keep making and releasing these short little pieces of RNA while the polymerase is still here bound to the promoter. And then you have scrunching happen. Scrunching is when it's pulling this DNA, the downstream DNA, into the polymerase interior. Eventually, you're going to stop abortive transcription and start making a piece of nucleotides that's longer than a piece of RNA that's longer than 10 nucleotides. Once you've gotten longer than 10 nucleotides, the interaction between the RNA polymerase and the promoter sequences is weakened, so that RNA polymerase is able to escape from the RNA binding sites and start moving down. It can release a sigma factor and just keep moving along the DNA molecule, adding pieces of RNA as it goes. Okay, so now all we have left is to talk about how we elongate and how we terminate, and then how we're going to process that RNA after we've made it, especially in our eukaryotic cells. What kind of processing lets the RNA leave the nucleus and go on for translation. All right, I'll see you next time. Bye.